Hello, everyone, and welcome to today's webinar, Insights to Heart Development and Disease Using Single-Cell Analysis and In Vitro Modeling of Pluripotent Stem Cells. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Sony Biotechnology. To learn more, visit them at sonybiotechnology.com. Now, we encourage you to participate today by submitting as many questions as you have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. I'd now like to welcome our speaker, Dr. Peter Andersen, an assistant professor as a group leader at Johns Hopkins University School of Medicine and co-founder of Vita Therapeutics. For a full biography of our speaker, please click on the Biography tab at the top right of your screen. Peter, welcome. You may now begin your presentation, sir.
Right, thanks for that introduction. My name is Peter Andersen, and I'm an assistant professor at the Division of Cardiology at Johns Hopkins School of Medicine. I'm also a founder of Vita Therapeutics, which is a biotech company that focuses on developing iPS-based therapies. Today I'm going to talk a little bit about how we use pluripotent stem cells as a model system to study some of the fundamental aspects of cardiovascular development and disease. My research is largely focused on early heart development and congenital heart defects, with a particular focus on congenital biology. My lab, we combine genomic approaches with high-content imaging and flow-based technologies such as flow cytometry and FACS to study these multicellular events in complex tissue down to a single-cell resolution. So for this webinar today, I'm going to briefly go over the current approaches to differentiate pluripotent stem cells with an emphasis on the cardiac lineage and cardiac differentiation. And then I'm going to go over some of the approaches to use pluripotent stem cells to study early heart development and demonstrate how we are able to recapitulate and model events associated with chamber-specific abnormalities in this system. And finally, I'm going to go over an in vivo-based approach where we develop a maturation system to mature embryonic cardiomyocytes, and which allow us to study age-related cardiomyopathies that manifest during adolescence and adulthood. So, many of you here are probably very familiar with pluripotent stem cells, so I'll just briefly go over this slide. When we talk about pluripotent stem cells, we generally refer to embryonic stem cells and induced pluripotent stem cells. Embryonic stem cells are typically derived from the inner cell mass of the blastocyst, which makes them ethically challenging to work with, whereas induced pluripotent stem cells are generated from somatic cells that are reprogrammed back to a pluripotent state by introducing the so-called Yamanaka factors. Now, iPS cells, they offer great therapeutic potential because they're derived from the patient's own cells. They're genetically matched to that patient, so they can eliminate tissue matching and rejection by the patient's immune system, which is a common problem that hinders successful transplantation. iPS cells are also a valuable research tool for understanding how different diseases they develop. So together, both embryonic stem cells and iPS cells, they have the capacity to self-renew and to develop into the three primary germ layers: ectoderm, endoderm, and mesoderm, and eventually into all the cell types that we have in our body. These differentiated cells can then be used for everything from disease modeling to validation of drugs in efficacy and cytotoxicity studies, which is now commonly used in the biomedical industry when evaluating new compounds in vitro. So since the first human embryonic stem cell lines were derived from human blastocysts in 1998 by Thompson and colleagues, there's been a tremendous interest in using iPSCs to study stem cells to develop both disease models as you can introduce mutation into these cells, as well as to potentially regenerate damaged tissue. The way we started to culture and differentiate these cells was by generating cellular aggregates that we kind of termed them embryo bodies, that you can see here. These embryo bodies were then differentiated using protocols that was based on spontaneous differentiation, and this spontaneous approach was a very efficient approach, as typically you're only able to develop about 1 to 3% of the cells that you're interested in this system. However, this way of differentiation was still the gold standard as late as in 2000, I would say 2010. So right around that time, there was a switch in the field where labs started to use more, they got rid of the serum-based protocols and started to use these defined serum-free medias. And doing that, and when we also at the same time followed the principle of development, we started to generate differentiation protocols that relied on stepwise differentiation using either cytokines, small molecules, to activate and block signaling pathways to first differentiate the cells into the 3 germ layers that I just mentioned, and then towards specific lineage of interest, again, by following the principle of development, hereby dramatically increase these protocol efficiencies. And nowadays, some of these protocols, or many of these protocols, they're in fact so efficient that they can robustly yield more than 90% of the cell types of interest. So because there was such a big interest in differentiating only the specific cells of interest, most of these protocols, they were also monolayer based, as you can see here. So that was around that time that all protocols started to kind of become monolayer based protocols. But then within the recent couple of years, there has been a push back to the 3-dimensional protocols again, where more and more labs are starting to focus on organoid technology. And basically, organoid technology, they provide more cellular complexity to the system, which often better reflect a given organ's properties, and thus making them more useful and relevant as disease models. And as these models, they can be used in validation studies of drugs and cytotoxicity studies, etc. So when we talk about cardiac differentiation, many of these protocols, they're very similar when it comes to differentiate mouse as well as human pluripotent stem cells. The only thing that really differs between the protocols between mouse and human is actually the timing. Most of the cytokines or the small molecules we use to differentiate these cells, they are the same. And the most common method of differentiation are induction of mesoderm by temporal activation of either Wnt signaling, typically through inhibition of GSK3 beta with a small molecular compound, or by co-treatment with the cytokines actin A and BMP4. Both of these approaches, they push pluripotent stem cells towards the mesodermal fate, which can be evaluated by analyzing the expression of some of the genes that I highlight here. As cells, they then start to commit to the cardiovascular lineage, and they differentiate into progenitors, which is that stepwise process. Then at some point, if you're interested in making myocytes, inhibition of Wnt signaling seems pretty crucial. So when you look at most of these protocols, you see that they start off by an induction of Wnt, and then later on inhibition of Wnt. So when we do this inhibition of Wnt, we force the cells into the cardiomyogenic lineage. And as with the cells we differentiate from the pluripotent stem cells and the cardiomyocytes that we generate, these cardiomyocytes, they unfortunately do still remain embryonic. That means that they resemble a state that is similar to the state of cells that we, or cardiomyocytes, during this late gestation in the developing fetus. At this point, we are now able to produce different subtypes of cardiomyocytes. And there's a lot of research going into this, but some of the interesting research has shown that if you manipulate retinoic acid pathways by either adding retinoic acid or blocking the retinoic acid pathway, you can push the cell towards an atrial versus ventricular fate, and you can also make changes to the protocol, so you can potentially also push the cells into these seno-atrial node cells. All right, so for this next part, I want to talk, I want to show you how we are using the pluripotent stem cell system and how we have used it to develop a model system to study heart field specification and development of the individual heart fields. So just to give a quick idea of why we're doing this, so early heart defects are a frequent source of mortality, estimated to account for as many as 30% of embryos that are lost before birth. And almost 1% of newborn babies that are born with a form of congenital heart defect, which actually makes congenital heart disease the most frequent type of birth defects in humans. The pathogenesis of congenital heart abnormalities is largely unexplored, and one of the reasons for that is due to the embryonic origin and the inability to obtain tissue from patients, which kind of makes sense. So in mammals, the heart develops from two sets of molecular and phenotypic distant progenitors, referred to the first and second heart field. And as you can see here, this drawing, the first heart field is visualized in red, whereas the second heart field is visualized in blue. And as you can also see here, the first heart field gives rise to certain parts of the heart, whereas the second heart field gives rise to other parts of the heart. So the first heart field gives rise to the left ventricle and part of the atria, whereas the second heart field exclusively gives rise to the outflow tract and the right ventricle. And it makes sense that abnormal progenitor development is closely associated with the etiology of congenital heart defects. So one of the questions we asked ourselves was whether it was possible to develop a pluripotent stem cell-based model system to study heart field specification and potentially allow us to model progenitor development and chamber-specific events that are associated with congenital heart disease. So as I just showed here in my previous slide about the first and second heart field, the heart is comprised of these two heart fields. So what we did for the study, we started out by visualizing and monitor heart field specification in vivo. And to do that, we analyzed the expression domains of several known genes that have been associated with the first heart field and the second heart field, and we used the mouse to do that. I'm not going to show you all that for the sake of time of this presentation. I just want to show that we did identify two genes that literally marks the first and the second heart field, as you can see in these developing mice. So here we have developing mouse embryos at 8 days post-fertilization and 9 days post-fertilization. And what we can see here is the first heart field is marked by GFP, which is driven by the gene HCN4. So HCN4 was a gene that is solely expressed in the first heart field, whereas TBX1 is exclusively expressed in the second heart field and is driving expression of RFP. So what you can see here on these pictures to the left is that these two heart fields can be identified in this mouse model, and it's in a non-overlapping manner. So it's a really nice tool to basically follow these progenitors throughout their development and in building the heart in these mice. So for us to develop a study in vitro or in the pluripotent stem cell system, we then next rederive embryonic stem cells from these mouse crossings with the correct phenotype, and thereby, providing us with the capability to potentially monitor first and second heart field development in vitro by literally evaluating GFP and RFP expression. So the first thing we did was we devised a differentiating protocol based on the embryo body technology, meaning basically cellular aggregation, to generate a 3D environment to resemble the in vivo environment as much as possible. And as you can see here, once we form these embryo bodies or spherical clusters, we then subject them into a differentiation protocol, as I was explaining earlier, where we are inducing the mesoderm induction, in this case by treating the embryo bodies with activin and BMP. And then at a later stage, the question was, are we going to see heart field specification in these? And we were hypothesizing that we were going to see GFP and RFP as a way to identify the first and the second heart field, and this is exactly what we did. So when we look at the picture here in the bottom, we see that after we've driven this induction by actin and BMP, see over time we start seeing formation of these separate domains that are either RFP and GFP positive, and they're adjacent to one another, very much similar to the in vivo scenario, where we see the two heart fields developing adjacent to one another. So the next thing we did was to first identify which of these concentrations or which of these methods gave us the best induction of cardiogenesis. So as you can see here, this is where we test out an increasing dose of BMP, as you can see in the top, and then also we have an increasing dose of activin A, as you can see out to the right. And then we're doing a scatter plot here of all of cardiovascular T here. So we picked out the treatment, or well, the condition that gave us the most amount of cardiovascular T positive cells here. So cardiovascular T marks cardiomyocytes, and as you can see, the condition highlighted in red is the one that gives us 36%. This is just an immediate time here that we pick that condition, and then at the same time we evaluated the expression of GFP and RFP. So when we look at this condition here, we can see here that that GFP is at the y-axis and RFP is at the x-axis, and we can see that we get a nice formation of GFP positive cells, as well as we get a nice formation of RFP positive cells. So that kind of indicates that we are actually forming first and the second heart field progenitors in this system. But to make sure that these are also, or to validate that these cells are actually also cardiogenic, we then cultured these cells a little longer, and then we analyzed the expression of cardiovascular T again. As you can see out to the right, we isolated the GFP positive cells, and we could see that 89% of them were actually expressing cardiovascular T as a sign of being myocytes, and whereas about 50% of the cells expressed cardiovascular T in the RFP population from the second heart field. And this goes very well with the literature, because in the literature we already know that the first heart field is primarily a unipotent cardiomyogenic heart field, meaning this heart field doesn't only give rise to cardiomyocytes, whereas the second heart field is a multipotent heart field that can give rise to different types of tissue or different types of cell types in the heart. So it makes sense that 50% of them are myocytes. And we actually have more data showing that it also gives rise to the vascular lineage smooth muscle cells as well. I'm not going to show that here for the second time. So the next question we had was, how similar are these cells generated in vitro to their in vivo counterparts? So to answer this question, we performed RNA sequencing on GFP and RFP positive cells in vivo as well as in vitro. This RNA sequencing analysis revealed a high correlation between in vivo and in vitro cardiovascular, literally telling us that there's a very similar gene expression profile between the two states, both in vitro as well as in vivo, and giving us confidence that the system actually works and we are generating first heart field progenitors and second heart field progenitors. So in that process, we identified approximately 1,500 genes that was differentiated and regulated between GFP and RFP positive cells. And of these, about 75% of the cells were enriched in both GFP and RFP positive cells, suggesting that our spheroid system really recapitulates early heart field development and specification. So we next asked whether we could use the system to identify pathways that are involved in the specification and development of the individual heart fields. To do this, we did an engineering pathway analysis, also called an IPA analysis, of these enriched genes, and we identified relatively few pathways that were differentiated and regulated. So as you can see here, the green bars, they represent first heart field cells, or GFP positive cells, whereas the red bars, they represent second heart field cells, RFP positive cells. So the higher activity of actin, cytoskeleton, and paxillin signaling likely reflects the presence of these structural genes being transcribed in the first heart field. So what we focused on was the actual pathways of BMP and Wnt signaling. Because as you can see there, BMP pathways was highly upregulated in the first heart field versus the second heart field, whereas the Wnt signaling pathways were highly upregulated in the second heart field versus the first heart field. So this tells us that somehow these two different pathways, they play specific roles within the individual heart field. So in order to test that, we tested whether BMP and Wnt signaling, they actually regulate specification of the first and the second heart field in our pluripotent stem cell system. So first we analyzed the effects of BMP signaling, and as you can see here, we find that BMP induced specification of GFP positive progenitors in a dose-dependent manner. And then when we did the same study and we tested out the canonical Wnt molecule Wnt3a, we could see that increasing dose of Wnt3a actually resulted in a dose-dependent manner of second heart field cells, but didn't affect formation of the first heart field. And what was really interesting was that when we treated these cells with BMP alone, we see formation of first heart field cells and second heart field cells, indicated by the green and red bar out to in the bar graph to the far right. And then when we add in Wnt to that system, we see that we're actually not affecting the first heart field, but we're increasing formation of the second heart field. And at the same time, when we block Wnt secretion, and this can be done by adding the porcupine inhibitor IWP2, we actually see that we're still seeing formation of the first heart field in presence of BMP4, but the induction of the second heart field is dramatically diminished. So literally, that kind of takes us to the current working model that we have, is that we worked out in this paper that BMP signaling drive formation of the first heart field, but at the same time, first heart field secrete Wnt that drive formation of the second heart field, and that's actually what we're working from now. And I'm just gonna move over and explain a little bit more about how we use the system to identify a surface marker to also identify these cells. So when we looked again into our array data or our RNA-seq data, we wanted to see if we could identify these progenitors by the expression of surface receptors. So one of the surface receptors we found that was highly upregulated in the second heart field progenitors was a gene called, or a surface expression called CXCR4. CXCR4 is a well-known receptor that is involved in migration of certain progenitors, and we found it interesting that it was highly upregulated in the second heart field progenitors, but not in the first heart field progenitors. So we turned back into our system, and this time we used another pluripotent stem cell line, this time being driven by the cardiac progenitor marker islet that drive RFP. So we can take these cells and we analyze them for CXCR4 expression, and what we actually find here is that within that population of progenitors, we can identify a population that expresses CXCR4 and one that is not expressing CXCR4. So when we take these two populations and we actually test them in clonal assays by qPCR, we can see again that the population that is not expressing CXCR4 is largely cardiomyogenic, as we would expect from progenitors from the first heart field, whereas we see as the cell type that expresses CXCR4, which we believe are the second heart field progenitors, it's more like a multipotent cell type, giving rise to all the variations of the heart. So next, we also want to show whether we could actually develop this system for human iPSCs. So as you can see here, we devised a very similar protocol. This time it's human with no transgenic markers at all. But because we have the information of CXCR4, we believe that we could use CXCR4 to potentially identify first and second heart field. So in the human system, we're taking human iPS cells, we're forming these aggregates, and we're differentiating them very similar to the mouse cells that I just mentioned. And at the right given stage, which is about five and a half days after we start the protocol, we try to see whether we can see a difference in terms of CXCR4 expression. And as you can see here down on the scatter plot in the lower left, see that there actually is a population that expresses CXCR4 and a population that doesn't express CXCR4. So when we sort these two populations up, you can see that both of them are very cardiogenic, which is expected as it is a cardiogenic protocol. In both cases, where about more than 70% of the cells are expressing islet, which is the transcriptional marker of cardiac progenitors. But when we then analyze these cells for the gene expression, we see that CXCR4 negative cells seems to have high enrichment of genes that are associated with the first heart field, ATN4, TBX5, and GATA4, whereas the CXCR4 positive cells is the opposite. They do not express these markers, but they have expressed the markers that are associated with the second heart field. So exactly what we were expecting. And as you can see out to the right, we see that, again, the population that is CXCR4 negative is largely cardiomyogenic, whereas the population that we believe refers to the second heart field is a little less cardiogenic. So it makes sense to show that our system can be used to also isolate the first and second heart field in human iPS cultures. So, next, I wanted to talk a little bit about how we also are able to use this system to model change-specific disorders using pluripotent stem cells. So As you can see here on my next slide, one of the first genes we want to events that we want to look at was the Holt-Oram syndrome, which is caused by a mutation in the gene called TBX5. So in the animal model, or in the mouse, what happens when we lose expression of TBX5, we end up with a very underdeveloped left ventricle. And this makes sense because TBX5 is very important for development of the first heart field. So this is where our system potentially could show us where we, if we're able to use this to specifically pinpoint whether there's mutation, whether genes are affecting either the first or the second heart field. And as you can see here in this assay, this is a pretty simple assay where we are differentiating our spheroids, and we then knock down TBX5. And as we can see here is that knockdown TBX5 dramatically reduces the formation of cardiomyocytes in the first heart field, but it doesn't affect the second heart field. So literally showing that we can actually use this system to study these chamber-specific events. And as another example here, we have the gene TBX1, which is actually one of the genes that we use to identify the second heart field. But deficiency of TBX1 is actually linked to a condition called DiGeorge syndrome, and which is associated with abnormalities of the outflow tract. So in this case here, deletion of TBX1, we would expect that it would only affect the second heart field because the outflow tract is derived from the second heart field. And this is exactly what we see. We see here that when we delete TBX1 in the first heart field of the DLP positive cells, we don't see any statistical difference between knockdown and not, whereas when we knock down TBX1 in the RP positive cells, we see a dramatic reduction in proliferation of these cells. So this again confirms that the system can be used to model these chamber-specific events. And just lastly, I want to go another study which is actually done with a collaborator in Copenhagen, where they had a project where they were evaluating mice that was deficient for a gene called SMERF1. And what they had seen in this in their mice was that it seemed that they had an abnormal outflow tract development. So again here we tested our system and deleted SMERF1 in our system, and we would expect only to see an effect on the second heart field. And this is exactly what we see here. We see that when we delete SMERF1 in this system, we see that it only affects the cardiomyogenesis in the second heart field, but not the first heart field. So again, this kind of validates this whole system and really excited us because it has given us a tool to study these chamber-specific events in a pluripotent stem cell system. Right, so before we move to the next part of my talk, where I demonstrated how we use the pluripotent stem cell system to identify a role of Wnt signaling in formation of second heart field progenitors. I want to use these few slides to show how we use the mouse model to validate our findings from the pluripotent stem cell system, and how we use the Chromium platform from 10x Genomics to investigate the impact of Wnt signaling during heart field specification and formation of the embryonic heart. As you can see here, we have here mice and basically it's HCN4 GFP, so identifying the first heart field. So you can see here that the left ventricle is expressing GFP because that's derived from the first heart field. But in this case here, what we did, we deleted a gene called Wnt, and Wnt is responsible for cells to secrete Wnt. So when we delete Wnt, that means that cells can no longer secrete Wnt. And what we see here is that in our control mice, we get a normal heart where we see the outflow tract and the right ventricle. They're not GFP positive because they're derived from the second heart field. And out to the left, you can even see that you have the RV, and it's not expressing the first heart field marker TBX5. But when we then look at the windless mutant embryos, we now see that we don't see a formation of the right ventricle. And furthermore, we can see that basically we only get one ventricle, and the entire ventricle is TBX5. So literally showing that we are not able to form the second heart field in these mice. So that kind of ties back to what we found in the system, was that that specification of the second heart field relies on formation of the first heart field to secrete winds. So when we don't have the first heart field being able to secrete winds, we are not able to induce the second heart field. So we next here employed a multi-sequencing approach where we used MESP1 to drive expression of TD tomato to identify the mesodermal derivatives in these mice. And we then had windless deleted, as you can see here, up to in the far left corner. So the stages that we analyzed was E8.0, E8.5, and E9.5. And we then, after that, we barcoded the cells using the 10x Chromium platform, and then sorted the cells so we can isolate the cells that derive from mesoderm, as only those cells are RFP positive. When we take these cells, we create these UMAP plots. We can identify all the structures within the mesodermal lineage here. Now, one thing that we're very interested in was obviously to look into the Luen pathways. So, as you can see here, what we identified here was actually only a subset of Wnt that was actually expressed within the mesodermal portion here. And what's kind of striking me is that the first heart field, they express high levels of Wnt2, which actually corroborated our previous findings, while the posterior second heart field expressed only low to detectable levels of Wnt. Now, the outflow tract cardiomyocytes, they highly expressed Wnt5A and Wnt11, which are actually both known for their roles in outflow tract development, but they're also both members of the non-canonical Wnt family. So this data actually further supports our hypothesis that the first heart field may be the source of the instructive Wnt that are specifying the second heart field. So we then here performed a trajectory reconstruction of control and knockout cells in the second heart field using monocole. So by using monocole, we reconstructed a branch trajectory in which controls and knockout cells are largely indistinguishable here at 8.0. Before we can see that they actually reach these separated states at later time points. So we subsequently used this trait sheet to identify genes that are differentially expressed across the control and knockout path at a branch point, focusing on genes with differential expression at the end stage. So here we identified around, I think it was 750 genes, which we classified into 4 clusters based on the gene expression dynamics. So in general, GO terms relevant to heart development were enriched in upregulated gene clusters shown out here to the right, and while GO terms related to cell cycle were enriched in downregulated clusters. And just to further show that, we can look at some of the individual genes, and what we generally see is that genes, as I just mentioned, that are associated with the cell cycle are kind of downregulated here, whereas we see that that genes that are associated with cardiomyogenesis tend to be upregulated in the control cells. I'm sorry, in the mutant knockout cells. And what we can further see out here in the embryos that have been treated with EDU, we can see that there's a drop in proliferation within the second pharyngeal arch, just demonstrating that the progenitors within the second pharyngeal arch have lost their proliferation potential. And what we were able to conclude from this study was that premature differentiation of second heart field progenitors and decreasing proliferation as a key mechanism for this knockout heart phenotype. So this is all in all, that kind of shows us what we were able to do. We were able to identify a role for Wnt signaling in the pluripotent stem cell system in this new model that we have created, and we actually end up validating in the mouse system to actually show that we're able to answer some of the specific questions in terms of Wnt signaling and their effect on early heart formation. So just to finalize this for today, I'm just going to spend a few more slides on talking a little bit about maturation and how we developed a system to study in maturation in human myocytes to study these adult onset diseases phenotype that we see. So, as I mentioned on one of my earlier slides, the cardiomyocytes that we generate from either induced pluripotent stem cells or embryonic stem cells, they retain these immature characteristics. I would say they're fetal-like, and they kind of stuck in that stage. There's a lot of push to get these cells to mature, but I don't think anyone has really identified some factors that can get them into the final mature state or adult state of the myocyte. And when we look here at the image here, we can see that a typical myocyte that developed from pluripotent stem cells is a small round cell, very disorganized in terms of the sarcomeres. Whereas when you look at the picture in the far right corner, we see that a true adult cardiomyocyte is an elongated cell, rectangular shape, with a very nice organized structure. So what we did for this study, we were asking, so what is it that makes the cells stop the maturation? So basically, they seem to be stuck in a state. And what we had already seen from other studies, and we'd also done some of these studies ourselves, is when we took these embryonic cardiomyocytes and injected them into infarcted models or adult hearts, we saw that they could engraft, not very efficient, but the embryonic cardiomyocytes, they did engraft, but they remained and retained their immature characteristics. They remained this inorganized structure. They didn't have a proper sarcomere alignment. So we then asked ourselves, so what is it that makes the difference? And we started to think well, what if we potentially inject cells at an earlier stage? Because we know that cells from birth are maturing throughout the adulthood. So we tested this hypothesis of injecting embryonic cardiomyocytes into newborn rats. And so basically, here's the scheme for this project. We differentiate embryonic stem cells or iPS cells in using the protocols that I explained earlier. And then we take these cells, we inject them into the heart of newborn rats, and we wait 4 to 8 weeks until that the rat actually reaches adolescence or even become an adult, and the heart has properly matured. So surprisingly, what we see here and what we found was that when we did this, and this didn't matter whether we injected rat ES cells or whether we injected mouse embryonic stem cells, we saw that these injected embryonic cardiomyocytes, they were able to undergo full maturation within the host, within the actual heart. So what we can see here is that the cells that we were able to extract from these animals when they were at an adult stage were highly organized and resembled all the characteristics of an adult cardiomyocyte. And when we quantified their maturation states in terms of nucleation status, it's well known that cardiomyocytes, when they mature, they undergo a process called binucleation. We see that these mouse embryonic stem cells or embryonic cardiomyocytes that we injected undergo binucleation very similar to the rat host cardiomyocytes, indicating that these cells are actually undergoing full maturation. We then performed a numbers of different assays to actually single cell based assays to show that these cells, they actually have all the functional probabilities of an adult cardiomyocyte, and to a large extent we see that they resemble or more or less indistinguishable from adult cardiomyocytes from the rat. So we even tested that with human iPS-derived cardiomyocyte, and we see the same thing where we see a complete matured state of a human cardiomyocyte here. So when we again did some single cell based on AC, and here we show that the PCA plot, we can see that the cells that we have matured or in vitro, meaning that they're not undergoing proper maturation, they separate out from the cells, from the adult cells or the adult cardiomyocyte, as well as the cardiomyocyte that we have matured in vivo. You can see that clustered together here, nicely highlighted as blue and green dots. So what we were really interested in here was that now it seems that we could mature these cells into an adult-like stage. So the question was whether we could also then use the system to study these adult onset diseases. And in this case here, we were very interested in arrhythmogenic right ventricular cardiomyopathy, which is an inherited form of cardiomyopathy that manifests only in adolescence or adulthood of humans. And it's largely characterized by a fibrofatty replacement, apoptosis, and intercalated disc abnormalities. So what we see here is when we take the cells and we kind of mature them in vitro, you see that the cells, they retain their immature characteristics, and they don't express any markers that are associated with disease. And that can see up here in the far left corner. The marker that I'm referring to here is pyrrolipin. We see no expression of pyrrolipin. But once we take the cells into our in vivo maturation system and we let them mature here, we can see when we do this with wild type or healthy iPS cells, we don't see expression of pyrrolipin, which is what we expect because these cells are from a healthy individual. But when we take cardiomyocytes from an ARVD patient or iPSC-derived cardiomyocytes, we see that they could correlate with pyrrolipin expression, something that we've never seen in vitro. So just suggesting or demonstrating that the cells are actually undergoing maturation as we would expect. And when we look at some of the other parameters, we see that there's a higher percentage of these cells, they tunnel positive as a sign that they are undergoing apoptosis. And finally, we also looked at the intercalated discs, and we can see here when we look at the normal human control, we see like this nice thin intercalated disc, which is a normal condition. But when we look at the ARVD cells that we have matured in our system, we see that there's a very big discrepancy in these cells. They look kind of off when you look at these integrated discs. So just to summarize that up, the last bit that I was talking about here was our in vivo-based maturation system of mouse embryonic stem cells or human iPS cells, where we can develop the cells, we can inject them into newborn pups, and in a matter of like a month or so, you can have these fully mature and adult cells. So with that, I'm just gonna stop right here, and I'll be happy to take questions.
Thank you, Dr. Andersen, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's take a look. We already have some incoming questions. Our first question, what are some of the challenges associated with working with the embryonic spheroid system in general, and specifically when sorting them for single-cell transcriptomics?
Yeah, no, thanks for that question. That's a great question. So I think, first of all, for working with this protocol and working with Pluripotent in general, it's all about reproducibility. So there's a lot of variability in this system. So you want to make sure you have some standardized and reliable differentiation protocols, and that also goes for this system. And that goes down to, make sure you plate the exact correct number of cells, et cetera, et cetera. So that's for that part. And then for the actual single-cell transcriptomics, so as I showed here, for some of it, we used the 10x platform. And it's generally all about timing. So you want to minimize time. So for the 10x protocol, that entitles that you have to be effectively barcoding the cells before you potentially sort them. And then for the other types of single-cell transcriptomics approaches, we typically use a 384-well plate system that we have already prepared with the lysis buffer. Then upon sorting, we will have a container next to the sorter, filled with dry ice, so that we immediately take the plates and flash freeze them right after sorting. And then, so typically from dissociation of the cells to the cells have been sorted and lysed, it shouldn't take more than 30 minutes.
Thank you so much. And did you use any specific approaches for sorting cardiomyocytes with high viability?
Yeah, that's also a great question. So one of the most important steps here is to have a good and robust dissociation protocol. So typically a lot of cells, they tend to die during this state when you actually dissociate them. So make sure you have a good, reliable dissociation protocol. Then after that, we use PBS that we supplement with heaps, typically around 10 millimolar. There's also other options out there for commercially available sorting solutions that somewhat increases the viability of cells. But to make sure you have a good sorting buffer. And as I said, it can be as simple as we do PBS without calcium magnesium that is supplemented with the heaps. That works for us. And then one important feature I'd say is that to make sure that the cells are cold at all time, so that when we use our sorting buffer, we pre-cool it. We make sure that everything is cooled. And then if you have a sorter that is equipped with temperature control, that will increase viability a lot. For example, we use the Sony sorter, the SH800, which has this feature. So I highly recommend you enable the feature if you have it on your sorter. That will significantly increase viability. And then finally, I would say, if you have different options for nozzle sizes, increasing the nozzle size will also increase variability because you lower the pressure on the system and on the cells.
Thank you so much. Our next question, what is the expected time in order for this technique to be used and the general practices, and what are the expected benefits?
So in terms of time, so I think, if we probably referring to the single cell sorting and the single omics that I just referred to. And again, I would say, you got to work efficient. You make sure, after you've dissociated cells, should take no more than 30 minutes or so until you actually have your cells ready or lysed for the next steps.
Thank you so much. And Peter, we have one more question coming in. This person says, my question is about how to maintain homogeneity of the generated CMs.
Oh, yeah, no, that's a good question. And if you saw my earlier slides, we can generate different subtypes of cardiomyocyte depending on what type of differentiation protocol you use, right? So you can differentiate them towards the direction of atrial myocytes or ventricular myocytes, and then obviously you can enrich these myocytes by certain sorting processes. But in order to maintain them in a homogeneous population, I don't think I have a good answer to that. Basically, the myocytes, when they're differentiated, they shouldn't really they shouldn't be that heterogeneous. So they should be able to just keep that, be locked into that state. So I don't really have a good answer to that question.
Dr. Andersen, I want to thank you for your time today and for your important research. And we'd also like to thank LabRoots and our sponsor, Sony Biotechnology, for underwriting today's educational webcast. And before we go, I also want to thank our audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and bye-bye.